Thank you very much, Mark, for giving me the opportunity to show you here what we are doing in Vall regarding rare diseases and specifically trying to find improved treatments for Fabry disease in this case. So I'm really sorry because initially my talk was going to be before the previous one from Minotics, and it was well planned in that sense because we are more in the preclinical part of a study, but I was attending a different conference now, so you will need to hear me after listening to these good results in the clinical levels. So just to start, I would like to know which is the background of the audience. How many of, I guess that most of you are working in lysosomal diseases, but how many of you are working diagnostic part of the diseases? None? All of you are working in therapy? How many of you are working with nanotechnology? None of you? Okay, so I will spend some few slides trying to show you what nanotechnology can do for the improvement of the therapy in the lysosomal storage diseases, specifically in those of them that can be treated with the enzyme replacement therapy. ¿Estás?
Yes.
Okay. So the idea here is that I'm coming from a group where we try to use nanotechnology to fill the gap between the basic research and the different clinical needs. We have different research lines. Most of them are based on the treatment of oncology, that, as you know, is one of the main fields in the nanomedicine. But at the very beginning, many years ago, we started working also with lysosomal storage disorders, and there is a reason for that. And the main reason is that most of the nanotechnological platforms, liposomes, polymeric micelles, whatever, they go straight to the lysosome. So we had a vehicle there to work with. So just to start it, as I said, I'm going to give you a brief introduction on nanomedicine, and then I will basically introduce you this European project that is called the Smurfo Fabry, where we're trying to develop these liposomes for the treatment of Fabry disease. So just to start with, as I said, we are working with nanomedicine, nanoparticles, and the idea here is that we are going to use this nanocyte carrier to act as a driver, as a vehicle for this active principle of cargo. So they can be very diverse in size and also in the constitution, but the important thing here is that this, using this nanocyte carrier to deliver the cargo, what we have at the end is a bigger structure than the active principle itself, and this difference in size and in composition is going to bring the nanoparticle and the active principle to a certain site. And not just that, because it's only a way to increase the solubility of the cargo and increase the bioavailability, because the nanoparticle is going to stay much longer in circulation and is going to reach a specific targets, especially if we are decorating the nanoparticle with targeting moieties. And this is going to be the case in the Smurfo Fabry project. So this is not science fiction. As you know, most of many nanoparticles and nanomedicine have been approved. They are already in the market. And here is just to show you that most of them, many of them, are actually liposomes. And if we can have this scheme out of the data from 2016, we can see that 20% of them are actually liposomes. And liposomes are nothing else than a small particle with a shell, a phospholipid shell, and this makes them extremely similar to the plasma membrane that is protecting a drug in the inner part. Usually this drug is an hydrophobic drug, but you can also use this system to vehiculize, to drive drugs that are not that hydrophobic. So there are also all the components that we can have in this structure that is going to make the whole system slightly different. One is the presence of polyethylene glycol, which is going to give some hydrophilicity to the whole system and make them more, let's say, less visible to the immune system. And also the presence of targeting system, as I said, targeting moieties, either antibodies or peptides in the surface that are going to increase the uptake from different cell lines, and also they are going to drive the nanoparticle to precise sites. So let's say that the main nanoparticle known in the field that is already approved in 1995 is this structure, doxil, which is a liposome that is pegylated, and it is also containing the doxorubicin in the inside part. So this has been the first approved nanomedicine in the market, and it has shown us how to how to handle the preclinical part of this type of system that are actually complex, because we are trying to work with different type of components, and it's not that easy from the clinical and preclinical perspective. Okay, so this is what we have. The nanomedicines are already there, especially liposomes, but how can we use them to treat the lysosomal storage disorders? So here we have a scheme of what is being actually used in the treatment of enzyme in the replacement therapy. And here what we have, just to introduce you the treatment, is the expression of the natural way of synthesis and biogenesis of the lysosomal enzymes, is that they are synthesized in the plasmatic reticulum. They go to the Golgi, where they get glycosylated, and then they are secreted to the extracellular media. So this is just part of the way. They can also go straight to the lysosome, but this is the most important part for us, because knowing that part of the enzyme is already secreted to the extracellular media means that we can also put a recombinant protein in the bloodstream and do the same process. The important thing here also is that this protein has to be properly glycosylated to be able to then internalize again in the cell and go to the lysosome. And this is actually what the enzyme replacement therapy is doing. It's putting the recombinant protein, and this protein can go inside the different cells through these mannose phosphate receptors. And as you can see, it's, the presence of the receptor and the presence of the proper glycosylation is really, really important to have the efficacy of the enzyme replacement therapy. In the case of using nanoliposomes or any type of nanoparticle to deliver the recombinant enzyme, here what we are going to do is that we are going to protect the enzyme from the degradation of the bloodstream, and we are also going to avoid the internalization through the mannose phosphate residues. We are going to use a different type of internalization, and depending on the targeting moiety that we use in the surface, we can increase the uptake for certain cells. And also, we can try to see if we can be able to cross the blood-brain barrier using specific targets. So this was actually the idea of the Smurfo Fabry project. We started with a nanoformulation of this type with, I will show it to you in a moment. And the idea is that we had some preclinical data supporting that this type of vehicle was going to be useful for improving the enzyme replacement therapy by lowering the clinical doses that could be used in the treatment of the patients, and by also reducing the treatment of the cost, because we were not going to be using a perfect enzyme, let's say. So we were at this stage, and the idea, was to have all the proof concept to support the translation into the clinical trials. And this is the members of the project. There are 10 different partners. Five of them are coming from research institutions, and 5 of them are coming from companies. So it's a pretty big consortium, as you can see here, and it's been tough to start working with it and organize, but at the end we are reaching to a point where we can really speed up all the research that we are doing in the field. So as I said, we're working product is this liposomes. They have 3 different components. They have in the membrane, in the shell, they have DPPC, which is a surfactant, and cholesterol. And the targeting moiety that I was mentioning to you is this cholesterol PEG with the RGD with a peptide at the end. This is inserted in the cell of the liposome and is driving the nanoparticles to those cells expressing alpha beta integrins because this is going to be like the natural ligand of those integrins. With this, the idea is that we can avoid the endocytosis through the mannose-6-phosphate receptors and target these nanoparticles specifically to the endothelial cells. As you know, lysosomal storage disorders are not specific for certain type of cells, but in the case of Fabry disease, there is a link between the presence of the GB3 deposits and the activity on the endothelial cells and the progression of the disease. So that's why we thought of using this RGD moiety in the nanoliposomes. Well, so one of the advantages of using these nanoliposomes and one of the strengths of the project is that to produce this complex system, we can do it in a single step. It's just by using, by putting different components in this pressured system and by using compressed fluids, we can just have in a single step the whole system form here. And this is something not just simple, but also easy to scale up under GMP conditions. And this actually was one of the objectives of the project. So these are the results that we started with, the nanoparticles, the nanoliposomes, when they did not have any targeting moiety and we were using them in vitro, we could see that they were not internalizing the cells, no fluorescent signal, but when we were adding this RGD moiety, the moiety that is going to bind the alpha-beta integrins, the internalization goes up. We have much more nanoparticles inside the cells, and we can also see that by these confocal images here where the targeted nanoliposome has a stronger signal, 30 times more internalization in these endothelial cells. Not just that, but they also go straight to the lysosomes. In this image, we have the lysosomes labeled in green, and you can see that the colocalization of the nanoparticles with the lysosomes really occurs here. But it's this really active. Are these nanoparticles able to bring the enzyme, the defective enzyme, into the lysosomes? And this enzyme that is being delivered is active once encapsulated, and the answer is yes. Here we have the results of the enzymatic activity. When we have the free enzyme, we have this activity around 300 micromoles per hour and milligram of protein. But when we encapsulate the nanoparticle, and this was very strange, we could not explain that very well at the very beginning, we have this increase in the activity of the enzyme. This is probably because, not probably, but we know now that the presence of the lipids there is stabilizing the activity of the enzyme, and that therefore we can have a higher activity. And when we translate that into cell culture system, we can see also that in the presence of the nanoliposomal system, we have a higher reduction of the GB3 deposits. The GB3 deposits are, GB3 is the substrate of the GLA enzyme, the enzyme that is defective in the Fabry disease. And when nanoliposomes are put in culture with these knockout cells with no expression of the GLA, this system is able to reduce these deposits, in a higher extent than the free enzyme. So in this case also we see that there is an important activity. But what is happening in vivo? And in vivo, we tested the pharmacokinetics of the nanoliposomal system. Here, encapsulating again the GLA enzyme, we could see that there is a higher plasma half-life and also a reduced accumulation in the liver. This was important because, as you may know, the presence of the mannose-phosphate receptors is really high, the expression is really high in the liver, and that means that when in normal situation, when you administer the recombinant enzyme to a patient, 60% of this enzyme goes straight to to the liver, and there's a poor bioavailability to other tissues in the body. But in this case, we were able to reduce this accumulation in the liver, but have more or less the same presence in the kidney, which is one of the organs that is the target of the Fabry disease, of treatment for the Fabry disease. So at this point, we needed, these were the results that we had when we started the project. At this point, we needed to optimize the nanoparticle, and this is going to, I'm going to show it to you in a moment. We needed also to introduce here a protein that was not going to be a protein that is actually available in the clinical setting. It was going to be a GLA freedom to operate. It means that the whole system would be able to be translated without the sponsor of a big pharma, in this case, because those were the owners of the clinically available GLAs, and they were not participating in the project. We also needed to scale up the synthesis. I will show it to you in a moment. Start with more standardized preclinical testing and complete the regulatory preclinical safety assess. So to start with, as I said, we needed to produce a GLA that was that we could have in enough quantities within the consortium without having the need to buy the commercial enzymes. And we did that. We had high activities in the batches that we were producing, even higher than the commercial one. And now, right now, we are obtaining the same version of the GLA that in the first version was having this his tag just to be able to purify it, and it was active, as you see here. And now we are just obtaining the first batches of the GLA without any CIBBIM tag in the sequence. So the next step was to introduce the GLA into the liposomes, in liposomes that were improved compared to the first version, because one of the problems that we had in the previous version of the nanoliposomes was that they were not able to encapsulate high quantities of the enzyme. The enzyme was active, but it was we didn't have enough quantities to start with the preclinical in vivo assays, and by adding to the complex this surfactant, the MAKC, we were able to increase the encapsulation efficiency and also the homogeneity of the liposomal system. The second thing was to change the targeting moiety, or at least add new targeting moieties to the nanoliposomes, and this was done by, one of the things was not to change the targeting moiety, but the length of the polyethylene glycol was used to link the RGD moiety to the cholesterol shell of the nanoparticle. And this is important because depending on this length, the nanoparticle is going to be different, and also the hydrophilicity that is going to have run is going to be different. And in this case, we were testing many different lengths, and what we saw is that a specific length of 400 was the best one to have both things, a nanoparticle that was stable and a nanoparticle that could work well in vitro. As I said, we also were testing different targeting moieties, not just the RGD, and these were the ones that we selected to be able to cross the blood-brain barrier. For the moment, there are two of them that are going to be analyzed, the one with the transferrin, and the one with the glutathione. I will show it to you in a second. And also, as I said, the scale up. In the previous results, we were working with a production that in a batch that where we were obtaining 7 to 10 milliliters in volume. Now we are working with a vessel of 75 milliliters, and they are also now building the whole system and having the first batches with these big vessels with up to 300 milliliters of volume. And this seems easy, but at the end it needs a lot of time and a lot of effort that was done in collaboration with, the work was done with Nanomol in the collaboration of Praxis, which is actually the owner of the patent of this system. The prototype of the pilot line for this big vessel in the GMP conditions is already available and is going to be built in the facilities of Praxis. For the regulatory testings, we are using also knockout models. The knockout model for Fabry mouse, no expression of the alpha-galactosidase A, and we are working in vitro using primary cultures and in vivo using the systems, the mice, just male mice, because as you know, this is an X chromosome-linked disease. And we've been doing different assays. I will show it to you, but before doing the efficacy assess, we started by testing the cytotoxicity and the hemolysis of the new version of the nanoparticles. We didn't see any effect, any cytotoxicity in the tissue cultures. We didn't see either any effect in the plasma coagulation times or in the hemolysis of blood samples. And we also tested that after repeated administration of high doses of the vehicle without any enzyme inside yet, we didn't have any side effects in vivo. So with this, we moved to see if the system was actually working. In vitro, we saw good results because, again, the nanoliposomal system, in this case with the MAKC, high quantities of the GLA, was able to reduce the GB3 deposits in the cells. And in vivo, we are just running the experiments now. Actually, we administered the mice 2 weeks ago, and we are waiting for the GBC results. The good thing is that the free enzyme is working as good as the alpha-galactosidase alpha, and I cannot show you the results right now, but it seems that the nanoliposomes are working even better than the commercial enzyme. We will need some more time to analyze the data. Regarding the preclinical testing of the systems with the peptides targeting the blood-brain barrier, they will be performed by Johanneum University, Johanneum Research in Austria. They have this specific system to test the blood-brain barrier crossing and the pharmacokinetics using this cerebral open flow microperfusion system. And they are actually doing this by using systems that are encapsulating fluorophores, just by measuring the fluorescence emitted by the samples that they are collecting from the brain of the rats in this case. And with that, the idea is that the last two members of the consortium are going to work in the preclinical regulatory assess. The idea here is to test the nanoliposomal system using rodent species, rats, and also beagle dogs. And they are just defining which are going to be the experiments and the assays, the doses specifically that they are going to be using, compared with what has been done already in different type of systems that have been approved for the treatment of Fabry disease. So with this, this comes to the end. This is the timeline that we have. We have completed some of the most of the preclinical assays with the old version of the nano-GLA system. Now we have to complete the testing of the system without MAKC. Initial results are good, but we need to repeat them and do more assays. And if with the blood-brain barrier assess, we see that the targeting system with the transferrin or glutathione is working better than the RTT, we may change the system and introduce those to have the dual targeting with the integrin targeting RTT and the transferrin or the glutathione. But we still need some time on that. The pilot plant, as I told you, is still prepared and being built in the facilities of praxis, and regulatory testing is meant to start at the end at the beginning of next year, actually. So with that, I hope I convinced you that the nanomedicine will be useful also for lysosomal storage disorders, and that in the Smurfo Fabry consortium, we are actually working in trying to see if this system would be useful for Fabry patients. And with that, I'm sorry there was a animation in here. I would like to thank all the members of the consortium that were in here, collaborators in here, and funding agencies at the back of this part. I'm sorry because I was using a previous presentation from Orlando. So with that, if you have any question or comment.
Thank you very much. I did not get the point how and why you want to reach the mannose-6-phosphate receptor with your liposomes. So
Okay.
when I understood you right, you integrated some integrins or integrin interacting proteins or
No.
what?
We are trying to target the integrins. To be able to target the integrins, we want to take the nanoparticle and drive it to the endothelial cells. So endothelial cells usually they have overexpression, not overexpression, the high expression of certain type of integrins. Integrins are heterodimers of alpha and beta, and there is this sequence of RGD, 3 amino acid, that is prone to bind these integrins, right? So we wanted to do this because we know that the endothelial cells and the stress of the endothelial cells is very important in Fabry pathophysiology. And we thought that maybe targeting and bringing this nanoparticle to endothelial cells could be helpful for Fabry disease. And that's why we were putting the nanoparticle with this RGD moiety. Okay, so this was one part of the issue. The second part is that we were aware that usually recombinant proteins of enzyme replacement therapy, they internalize through these mannose-6-phosphate receptors. And we didn't want to get rid of this type of internalization, but we wanted to have an alternative way of entry. Why? Because if you just rely in the internalization through the mannose-6-phosphate receptor, your enzyme is going to go to those tissues with high expression of this receptor. And other tissues with lower expression are going to have less presence or less quantity of the protein. And by using this alternative system of internalizing in the cells, we could have a better efficacy.
But before you reach the mannose-6-phosphate receptor, you have always to transfer or pass through the endothelial cells, where the mannose-6-phosphate receptor are very low abundant at the apical site. They are more localized at the basolateral site, and you have to transcross the cells before you reach the cells beyond or below the endothelial cells, right? So the liposomes, they give you the perhaps the advantage that they can fuse with the plasma membrane independent of receptors or something, and can be endocytosed with along the endocytic pathway directly to the lysosome. So did you test it whether your liposomes can be inhibited by mannose-6-phosphate?
Yes.
The uptake?
We didn't do the experiment. But the thing is that the internalization with the targeted system is much more higher than the internalization without the RGD moiety. So that's why we think that the internalization is specific, because the internalization of the nanoliposome by itself is really low. Right?
Okay,
But it was a good point. I can discuss this with you later. But the thing is that as I mentioned at the very beginning, this liposomal system is usually used for hydrophobic drugs. So in the case of a protein, we don't have an hydrophobic drug. We have something that is more, in this case, is not really hydrophobic. And what happens is that the GLA, the enzyme in this case, stays inside and also in the membrane. So when it's also in the membrane, it means that maybe some of the mannose phosphate residues are being exposed to the outside, to the outside of the nanoparticle, to the cells. So we cannot discard that there is an internalization also going through the mannose phosphate receptors. Because the initial assays that we were performing, they were done in the absence of the protein. So there was no protein in that. We need to repeat those by having the protein present and see if the internalization goes more through the integrins or through the mannose phosphate receptor.
Perhaps I have also to kill some of your illusions. I have to say that the recombinant enzymes, they do in the maximum contain 20% of mannose-6-phosphate residues per molecule. So every fifth molecule has perhaps a mannose-6-phosphate residue. So this does not play an important role. So during when you injected your liposomes and you said the majority goes to the liver and something. The liver is also not a very high content mannose-6-phosphate receptor organ. So you have an uptake via the mannose receptors and not on the mannose-6-phosphate receptors, only to keep this in mind. Okay. But my question is now, how much protein you can deliver in total in your liposomes? You know, the patients need between 0.5 milligram and 1 milligram, sometimes, depending on the enzyme, 10 milligram per kilogram body weight. Yeah. Can you apply so much with your liposomes? With
the previous version of the liposomes, we were able to deliver 1 milligram per kilogram to the animal model. With this newer version of the liposomes, we are able to increase that 5 times. So it's 5 milligrams per kilogram. So still is far from what you need, for instance, in Pompe disease, where you need to have 20 milligrams per kilo. But we don't know, because we haven't performed, well, we have performed the assays, but we haven't done the analysis yet. We don't know if your pharmacokinetics is better and you have a longer half-life, you may need to reduce the dose that you are administering to have the same effect. So this is one of our hopes, that we can increase the half-life of the protein in the bloodstream, and with that reduce the dose and the frequency of the administration. But we don't know it yet, yes.
Okay, any more questions?
Yes. I have one question about this in vivo data. About the in vivo data in mice models, as you mentioned, that I was wondering, have you already measured, or do you know what is the target bioavailability of the liposomes in the brain? How much of your dose actually comes into this?
So the crossing of the liposomes to the blood-brain barrier, we don't know yet how much is going to be crossing. How much will we need to be able to deliver, to cross, to be able to have a reduction in the GB3 deposits and have a clinical effect? It will be probably low, but I cannot give you the exact number. The thing is that in Fabry disease, there are neuropathological disorders, but they are not they are not the important part of the disease, because the patients are not that much affected. And also, if you take as an example the enzyme replacement therapy, you don't need to replace the whole quantity of the enzyme that a healthy person will have. Just by delivering a small portion of it would be enough.
No, so I was just thinking mostly about the nanocarrier, the cargo as you are using it. How much of your nanoparticles 1 million, if you inject 1 million, how many will get into the brain?
I cannot give you the answer. I don't really know. In this case, I don't know. For other type of nanoliposomes, if you are able, that 0.2, 0.3 of the dose, if that is able to cross the blood-brain barrier, they call it a success. So we still need to test it here, so we don't know. We know for other systems that that we are using not nanoliposomes but exosomes, for instance, that with small quantities we can have a reduction of the GB3 deposits. But with this system we haven't tested it yet. I'm sorry.
Okay. Thank you.
Okay, thank you very much.